As I said at the beginning, I think Suresh, I think has given the best title I've ever seen. That's not to do you down, Jack, but I really like the idea of anything to do with who shot Bambi. So Suresh, over to you to tell us about Bambi and COVID, and hopefully it killed the deer.
Thanks, Julian. I just have to make a disclaimer that I did not entirely come up with the title. There was a BBC Science in Action interview I did, and they called the segment as Bambi got COVID. So I just sort of added, oh dear, to that. So I have to give partial credit to BBC for that. Thank you for the invitation. Can you see my slides okay?
It's all good. We can see and hear you just fine. Thank you.
Excellent, thank you. For the purpose of this presentation, I wanted to start off with a very brief overview of coronaviruses and interspecies transmission, recognizing that this audience know about this topic very well. And then I will very briefly introduce some of the work that we've been doing on the question to answer susceptibility and potential adaptation of the virus to animal species. And then talk about the Bambi COVID story and why we went after Bambi and what we found. And then maybe touch on the implications to the long-term trajectory of the pandemic. So as we all know, coronaviruses live in natural hosts like bats or rodents, as we just heard from Jack. And the interspecies transmission could enable the virus to jump species and infect an intermediate host. And then a zoonotic transmission of the virus could potentially result in an epidemic or a pandemic. And consistent with this hypothesis that a virus that originated from a reservoir and infected humans is also likely to cause what is called the zooanthropogenic transmission, essentially humans to animals. And by now we know that SARS-CoV-2 has many hosts, although the exact origins are still being debated, but we know it's from a bat and has infected multiple animal species, and the number still is increasing. And there are natural infections in many wild animal species in captivity and also pet animals. And there were reports of many studies about the natural experimental susceptibility of some of the animals like cat and ferrets, and their ability to transmit virus to the members of the same species, so which is consistent with the proposed zoonotic origins of SARS-CoV-2. So my lab, since the start of the pandemic, we've been focusing on a number of aspects. So one of the key areas that we focus is the susceptibility and the adaptation of SARS-CoV-2 in animals. The studies are funded by the National Institute of Food and Agriculture, the USDA in the US. And we also have projects that look at development of COVID vaccines and diagnostic tools. The key aspect is that as the virus continues to evolve, there are more and more variants emerge. And it is also possible that as the virus changes, the ability of the virus to infect other species or become more efficient in infecting animal species is likely to change. And with that in mind, we have been working with colleagues in the chemical engineering department of Penn State to develop computational models that would allow us to investigate the potential changes in the ACE2 RBD and how they may change the virus's ability to bind to ACE2 of a given animal species. So again, I don't want to go into much of the details here, but the idea is that it uses a neural network molecular mechanics model. And these studies were published last year. And the advantage with this particular model is that we can computationally mutate one residue at a time and see how that might change the ability of the virus to infect or bind, I must say, to ACE2 of a given animal species. So we have some of these tools, including multiple serological assays, both species-specific and species-agnostic, that we have developed as part of the USDA project, including the serotype assays to look for a different spike binding of serum antibodies. And then the study that was widely publicized in the US and across the world is where we found extensive natural infection of white-tailed deer, first in Iowa. And I just wanted to give a background of why we went after white-tailed deer. So this study, which computationally predicted several species to be susceptible to SARS-CoV-2, has placed white-tailed deer on the highly susceptible category, so which is evidence 1. And the second evidence was there was experimental studies done by our colleagues in USDA that showed these animals are not only susceptible, but they also give the virus to other animals quite easily. And the third study was a serological survey conducted by USDA in 4 different states in the US, including Pennsylvania, where I am located, that found nearly 40% of the deer sera that they surveilled had antibodies to SARS-CoV-2, which indicated there may have been extensive natural exposure of white-tailed deer to SARS-CoV-2. So then we wanted to investigate the actual evidence, if you will, to look for the viral nucleic acid. And just for a background, these are the most abundant large mammal species in North America, nearly 30 million numbers. And they have a significant contribution to US economy through a number of activities, including hunting and other recreational activities. But for many people, it's also a very emotional aspect because they have had this history of hunting in the family. And for some people, hunting deer is the major source of their meat. And the map shows the distribution of white-tailed deer and the density among different states in the US. So to answer this question, we had major challenges in accessing samples, as you can imagine. So fortunately, we found a Department of Natural Resources in Iowa as a good partner. And they have been carrying out white-tailed deer surveillance for chronic wasting disease. And they would typically collect retropharyngeal lymph nodes for this purpose. So we came to know that they have retropharyngeal lymph nodes stored from white-tailed deer for the past several years. And that gave us an opportunity in 2021 to literally go back in time and see at the early part of the pandemic if deer in that particular state have been exposed. So what you're seeing on this screen is the map of Iowa with the public lands all outlined. And you'll see a series of black and red dots. So the black dots represent negative samples, and they are all temporally and geographically identified. So we know exactly which location the deer came from, including the GPS coordinates, and also the time of collection. And I must also say, these were submitted by either hunter killed or killed in a road accident. That is how typically the surveillance of chronic wasting disease happens in the US. So between April of 2020 through August, several samples that we tested, all of them were negative. You can see they're all in different parts of the map in the state. And in September, we saw 2 positive samples in 2 different locations within the state that are a couple hundred miles apart, which indicates that these are likely to be independent spillover events because deer are less likely to travel that far. And if you just zoom into one of the location, lots of negative animals and 1 positive animal, and then zoom into the other location, similarly, many negative animals, 1 positive tissue. And then as the time progressed into October, we saw 1 more animal. And by November, there are a bunch more. And by December, we saw a lot more samples. If you just zoom back and look at the distribution of positive samples across the state, there were a lot of them. And this was up to 300 samples by that time, roughly 283 that we decided to publish these studies. But now we have processed over 4,000 samples from the same period. And now the map that I still, we're working on that, but it looks a lot more, pretty much every part of the state had positive animal, and nearly 50% of the samples we tested were all positive. So it shows extensive, widespread natural infection of deer in Iowa. which sort of made them at that time the first identified free-living animal species outside of humans that was extensively infected. Interestingly, we wanted to see how the positive rate among the deer tissues sort of map to the cases in humans, although this is a retrospective study and it was opportunistic sampling, so it's not a properly organized epidemiological analysis. But however, if you look at this particular graph, the blue bars are positive cases per 100,000 population according to the public health records in Iowa. And the right side y-axis is the percent positivity among deer. So during the months of November, December, when there was a peak human infection, and that's also when we saw the peak of positivity among humans. Although the other complicating aspect is that also happens to be the winter and the hunting season that displaces the migration patterns of deer. And also that's the time when we get, or the Iowa DNR gets the most samples. So there are lots of confounding factors, however, but it shows that the peak of human infection also coincides with the peak positivity among the deer. And as most of you know, based on the 2 or 3 independent experimental studies so far, we know that deer experimentally infected do not show any clinical symptoms. And they can transmit the virus, and the virus can be detected, or the nucleic acid, in lithoropharyngeal lymph nodes for up to 21 days. And we did the whole genome sequence of the SARS-CoV-2 samples from the deer. And then we compared the whole genomes from human cases from Iowa at that time. And what you see is the lineage of SARS-CoV-2 we found in the deer nicely overlaps with the circulating lineages among humans in that time, which further suggests that the source of the virus to deer is likely coming from humans. And there were lots of questions about how this might happen, although we did not know, we still do not know fully because it was a retrospective study. But there were lots of different possible ways this can happen. Could be contaminated food source or environment, or there may be a possible intermediary or bridging host that we do not know yet. And then that was from the early part of the pandemic, October until January of 2021. And we wanted to continue to see what was happening early this year. So we partnered with the New York City parks, along with a conservation group called White Buffalo, piggybacking on an ongoing deer sterilization study in Staten Island. As some of you might know, this is the park in Manhattan, New York, which is close to densely populated human dwellings. And this study was interesting because the veterinarian that were capturing the deer for sterilization were able to observe the animals for any obvious clinical signs, but they also have collected matching serum sample as well as nasal swab from these animals. And what you're seeing here is the Staten Island map. The blue circles are all where the animals were captured. And this is looking for the viral nucleic acid. We found 8 of 71 animals positive for the viral nucleic acid. And when we sequenced, there were 4 Delta variant and then 4 Omicron variant positive samples we found. And the Omicron sequences mapped again closely with the circulating Omicron variants in New York City. So did not see a major genetic change in the viruses that we found, or the sequences, I must say, in the deer samples. And then we also did the matching serum samples from a much more bigger sample size, 135 animals. And 19% of the samples were positive for antibody by a surrogate virus neutralization test. And then we did pseudotype neutralization using all variants of spike variants. And a lot of them reacted strongly with the wild type B1 lineage and not the Omicron. And if you look at the antigen cartography, again, they clustered closely with Delta and Alpha and the B1 lineage, and away from Beta and Gamma and Omicron, so suggesting that there was an ongoing or prior infection of this particular deer herd with an earlier variant or earlier version of the virus. And then Omicron has been a recent introduction to this herd. And as we were studying these things simultaneously, our colleagues in the University of Pennsylvania have also found two alpha lineage deer SARS-CoV-2 samples. The sequencing suggested they are significantly diverged from circulating alpha lineage in Pennsylvania at that time, which indicates that the virus is evolving in these animals. And more recently, a study from Canada showed there is a significantly mutated version of white-tailed deer clade SARS-CoV-2, which is shown here, that also further suggests that the virus is evolving in the deer in North America. And I did not put it in the slides, but also wanted to mention simultaneously, USDA has confirmed infection of white-tailed deer in more than 15 different states in the US. So this is not just a story about Iowa or Pennsylvania, New York. It's pretty much all over the North America potentially. And now the question is, we also know that when the virus gets into animals, they are likely to also give it back to people. So we know the story of mink farms in Denmark, and then subsequently the Hong Kong incident of the Delta variant through the imported hamsters from Netherlands. And also the Canadian study suggested that there's one individual who they screen tested positive with the white-tailed deer clade. Although the suggestion is that this individual had a notable contact with the deer. Therefore, the conclusion of the study is that the most likely source of this particular version to that individual was white-tailed deer. But again, it's N of 1, so we don't fully know if that is the case or are there other sources of infection. But the summary is that the virus not only has widely circulating in the white-tailed deer, there is also a significant possibility that the virus deer might be giving it back to people. So then the bigger question is, if the virus continues to spill over into animals and circulate, and what are the potential implications? So again, as we all know, there are several potential hosts of SARS-CoV-2 based on natural evidence of natural infection or experimental infection, or computationally predicted that they might be susceptible. So, and the list continues to grow. And there are, for example, some animal species that also live in the same ecological space as the white-tailed deer, say in the US, like deer mice or bush-tailed wood rat. And you can quickly see how this can get really complicated. And this cartoon just to show how the whole transmission networks could be very complicated, and we may not fully know what is happening. While we know the farmed livestock are potentially less susceptible or likely not susceptible, but there are lots of animals that we saw in managed or captive settings like zoos or domestic or shops that have been infected with the virus from humans, and some of them gave it back to people. And the mink and white-tailed deer that also are in natural habitat that we know are infected. And the question is, who else they're giving the virus to, and what kind of transmission networks might have been established. And that also suggests that there could be multiple ways through which the virus can get back into humans, maybe as is or potentially mutated. And in particular, we have been advocating agencies in the US in several meetings with the government and other regulatory agencies to focus on more targeted surveillance of some of the high risk species. So, for instance, the feral cats that go back and forth between domestic dwellings and to the woods, they may be in constant contact with the white-tailed deer and deer mice, and you can soon imagine the complexity of virus transmission can be very difficult to monitor unless we are intentional about that. And that could also mean that the virus can continue to change if we do not pay attention to. And finally, that's why I think this audience again know very well the importance of One Health approach to tackling emerging infectious diseases like SARS-CoV-2. So we are focusing on the full picture than a partial part of the equation. With that, I just wanted to summarize that now we know SARS-CoV-2 infects a wide variety of animals, and there is now a strong evidence of widespread natural exposure of white-tailed deer in North America, and an evidence of a potential adaptation of the virus in these animal species. Therefore, they are likely to serve as a reservoir because they also could remain asymptomatic. And at least there are 3 documented animal to human transmission of SARS-CoV-2. And therefore, now I think SARS-CoV-2 is a multi-species zoonosis and should be dealt with the One Health approach. In particular, since we cannot surveil all animals in all locations, it is highly critical that the targeted surveillance of high-risk animal species is really necessary to better understand the ongoing evolution of this virus. With that, I would like to thank a whole bunch of people that have contributed, many of my colleagues from Penn State and the New York City parks, and the Department of Natural Resources in Iowa, and the White Buffalo, and funding from USDA Animal and Plant Health Inspection Agency services. And then our colleagues in Houston Methodist Hospital, who did most of the genome sequencing, and then the Iowa State Veterinary Diagnostic Lab. And we also collaborated with Andrew Conlon and his group at the University of Cambridge for some of the transmission modeling that I have not presented here. And Jim Davis has done lots of analysis of the sequences. And these studies were funded by generous funding from USDA and Department of Agriculture in Pennsylvania and the Huck Institute of Life Sciences. And as I mentioned, lots of media attention to these studies, including the BBC story and a documentary on PBS NewsHour. And then New York Times had a cover page with my picture with a deer called Cashew. This is a deer facility we have on campus. And these are bottle-fed deer, so they just come to you like your pet dog. So this is a boy. His name is Cashew. So I'll stop there, and I'm happy to answer any questions. Thank you.